Good morning, students. Today I am going to discuss about embryology topic. Usually, what is embryology is nothing but the study of embryo. That is the study of embryo, which is called as embryology. First of all, embryology was discovered by Panchran Maheshwari, scientist. He is the father of embryology. Now, usually in the flowers which are having five different type of the pods, like one that belongs to sepals and petals and calyx, even stamens and androecium. These are the four different type of the pods which is present in the flower, and these are mainly classified into two parts, that is essential and non-essential. And non-essential is nothing but these are the sepals and petals which is not participating in the fertilization process. Now I can explain about essential pods. These are mainly two types in the flower, that is nothing but stamen and androecium and ovary. These are the these are all which is belongs to female sex organs which is participating in the reproduction. And now I can explain in the deeply the male sex organ, that is nothing but stamen. This stamen, for example, I am taking one example, datura metal. And datura, this is the one of the medicinal plant and which contain the different type of the flower pods. Mainly the stamen which are having how many stamens which is present in the datura, which are having mainly five stamens are present. Then first stamen which are having mainly two pods, that is anther and filament. The filament tip which are showing anther pod. The next one, next based on anthers, the anther which are having rooms, based on the numbering of rooms, which is classified into anthers mainly two types, that is dithecous anther and monothecous anther. Dithecous anther, which means which is present only two rooms are present in the anther. Monothecous is nothing but which are having only one room which present in the anther. These are the main two different type of the anthers are present in the angiosperms. See, then now the in the gymnosperms, these this type of similar character of the flower is not present. The similar character of the flower, that is the cone, which are having male cone and female cone, and the male cone which are present in the different type of the leaves, that is the microsporophyll leaves. In the microsporophyll leaves which are present microsporangium. In the inner side of the microsporangium we can see the microspores. These are the male gametes, and after the development of the male gametes to ready to participate the fertilization process. In the similar with the female cone is also present megasporophyll leaves. In the megasporophyll leaves which are present megaspores, these are also called as ovules. After the ovule, the development of the female gamete which is participate in fertilization process. See, what is fertilization means? The fusion of male gamete with the female which is given the zygote is directly converted into embryo. See, after that embryo, after the germination of the embryo, which is giving either diploid plant body, that is nothing but sporophytic plant body. In our botany having two different type of the plant bodies are there, that is sporophytic and gametophytic. Sporophytic is nothing but diploid in nature, and gametophytic is nothing but that is nothing but only single set of the chromosomes are present. These are the gametophytic. Now I can explain the general structure of the anther. This is the common anther ts. We can see the like butterfly structure on the microscope, and which are having majorly three parts in the anther, that is epidermis, endothelium. So this is the first part epidermis, and second part endothelium, and third part middle layer, and fourth one that is the tapetum. Then inner side which present micro pollen grains. See the general structure. This is the general structure of the anther, which are outline structure that is similar with the butterfly structure. Under microscope we can see the different type of the pods, that is epidermis, and that is the first one epidermis and endothelium, and third one middle layer, and fourth one tapetum cells. The most important character in the anther, these are the pollen grains. Now I can explain in detail the anther layers. The first one epidermis, which is present to the outside, and epidermis which are having cells uniseriate without intercellular space, and each cell which contain nucleus and then cytoplasm. There is no chlorophylls. The chlorophylls are completely absent in the outside of the epidermis in the anther. This is a very important epidermis which is which are helping in the protection to the inner tissues, even the inner pollen grains. Now I can explain the second row, either the behind of the epidermis, which are showing one more layer, that is nothing but endothelium. The endothelium is where which are having 2 to 3 layers, and which are helping in the dehiscence of the anther. Why? Because the endothelium cells which absorb moisture, either water, from the environmental condition at the time of maturity of the pollen grains, the which are helping in the stomy of the anther, either a rupture of the endodermis. Now the third one, that is the middle layer. The middle layer which are having 2 to 4 layers, there is no particular function of the middle layer. Then inner side, a particular double layered the cells, that is called as tapetum cells. These tapetum cells are very important. Whatever in anther, in another internal structures, even the tapetum cells, the function is which is produce the food material, whatever water and minerals to the to develop the development of the pollen grains, the inner side. I mean it, the tapetum cells which are helping in the nutrition. And these are the common 4 layers which we can see under microscope: outside epidermis, and lower side endothelium, and third one middle layer, and fourth one, that is the tapetum cells. Now the inner side, you can see the small dot like structures. These are called as pollen grains. After the maturity, the pollen grains, we can see the pollen grains. See the pollen grains, inner side of the tissue of anther, which is called as microspore mother cell. The microspore mother cell, which contain 2 sets of the chromosome, which means which is participating only meiosis division. After that, the participation of the meiosis division, the microspore mother cell which producing 4 microspores. See, these are the called as tetraoids. See, one tetraoid which is participating further divisions, this is called as male gametes, the development of the male gametes. See, after that we can see a particular male gamete, that is called as pollen grain. See, these are all anther structure in the anther walls and inner side of the tissues, even inner pollen grain structure. Now I can explain the general structure of the anther in detail. Before now I can explain normal structure of the pollen grain. This is the general structure of the pollen grain. We can see in Hibiscus rosa sinensis. This is the common name Mandara, and showing like this structure. And this is the very clear visible under microscope. The pollen grain which is surrounded 2 layers, outer and inner layer. The outer layer which is also called as exine, and inner layer which is called as intine. See, the exine with made up of sporopollenin product, just a very important for MSc question. Then second one intine, the intine with made up of cellulose and hemicellulose. Next one, in the exine and intine we can see, we can observe the like spores, either pores. These are the germinated pores. The germinated pores which we can see the S-shaped structure, very important on the microscope. Exine and intine we can see the pores. These are the called as germopores. That these are the size S-shaped structures are present. Then what is the functions of the spore of germospore is nothing but the pollen tube germinated from these type of the spores. Next one, in the inner side we can observe in the central axis, you can see in the central axis, and also pores even in the cytoplasm, right? This is the common pollen grain structure. It's a very important in a hibiscus pollen grain. In the exine we can see ornamentals. See, Every pollen grain, for example, I can see every pollen grain like this structure which are showing like ornamentals, like this type of ornamentals on the surface of the hibiscus pollen grain. You can see the under microscope a particular and clear visible of the ornamentals. This type of ornamentals, either spines, which is present only hibiscus pollen grain. This is a very important character. Now this is the spines structure. These are the general structure of the pollen grain. So before that I can explain NPC system. So now I can explain the NPC system, either the pollen grain, either palynology. What is palynology? First of all, I can explain the definition: the study of pollen grain, which is called as palynology. See, the branch of the study of pollen grain, which is called as, the branch deals with the pollen grain, that is called as palynology. Right now, the palynology, the study of fossilized pollen grains, which is called as paleobotany. That is a very important botany is nothing but the study of fossilized pollen grains, either fossilized plants, that is paleobotany. Right now I can explain the pollen grain outer structure. Before that I explain, which is covered two layers like exine and intine, and here also there, and exine with made of sporophyllin product, and then intine which is made of cellulose and hemicellulose, and we can see the absorb the different type of the spores in the outside. See, the exine again divided into nexine and sexine. That is a very important. Now the pollen grain size, we normal the pollen grain size, which is declared by the using of polar axis and median axis. Based on the polar axis and median axis, the normal pollen grain size that belongs to only 5 μm to 200 μm. In between the 5 μm to 200 μm, we can see in different the pollen grain sizes differ by the species to species and genus to genus. Even now I can explain the shape is also based on polar axis and medium axis, and we can calculate the size. Usually the pollen grain size, we have a one formula: polar axis by E into 100. That is nothing but polar axis by using P by E and 100. This is a very important. I can show here, that is P by E into 100. This is the common formula for shape. Now I can explain, before that I can explain symmetry. Each pollen grain which are having a prominent symmetry. Why? Because when the pollen grain which is divided into in the similar parts majorly. See, that is also very important. And the pollen grain surface which are having different type of the pore like structures, these are called as germpores. The germospores which are helping in the process of germinative of the pollen tube. That is a very important. Okay, now I can explain a very important system, that is nothing but NPC system. You can observe this is the general NPC system. NPC means the pollen grain, N means number, the pollen grain, P means position, the pollen grain character. What is nothing but the pollen grain number and position in character. This type of pollen grain number, position and character first discovered by Headman scientist. So very important. NPC system was discovered by Headman scientist. Right, in the system of NPC, we can observe stream suffix every time we can use as stream. See, now I can explain the NPC system. First I go with number. See, so based on the spore number we can mention a particular name. You can observe a particularly atrium. See, atrium is nothing but this is the first one which has not have any pores, that is the atrium. Pores are completely absent, then that type of pollen grain is atrium. See, complete stream I mean pores, number of pore is completely absent. N1, the second one, N1, the number of pore is only one, which is located to the outside, that is monotrim. Monotrim is nothing but indirectly which are having only one pore is present. N2, N2 is nothing but that is ditrim. Ditrim, which means which are having which number of which two numbers are present, the two pores will present. Then N3, that is tritrim. Tritrim is nothing but which contain three germ pores are present on the surface of the pollen grain. Then N4 is nothing but that is tetra, which contain four pores are present on the surface of the pollen grain. Then next N5, N5 is nothing but these are the pentatrim. Pentatrim is nothing but which contain five germpores are present. These the five germpores which are present on the surface of the exine. Then N6 is nothing but exatrim, and polytrim is nothing but more than number of the six, either seven. So this is anemotrim is nothing but finally anemotrim is nothing but the number of the spores is infinity, which are arranged in irregularly. These are all belongs to number of germpores on the surface of the exine. Based on number, we can classify it into monotrim, ditrim, and tritrim, and tetratrim, pentatrim, hexatrim, and polytrim, and finally that is anemotrim. These are the number. According to advanced scientists, the pollen grain which are having a particular position, if there is no position, that is P0, and the second one, if the pollen grain, the germpore which is present to the lower side in the particular position, that is catatrim, right? Then second one, the pollen grain, the germpores which is present to the two poles, to out pole and lower pole, that is anacatatrim. Then anatrim is nothing but the pole which is located to the outside of the pole. This is the anatrim. See, I can, this is the anatrim. Now zonotrim, zono, which means which are present in the zonal side, in which is nothing but the trims, the pores which is located in the middle position, that is zonotrim. Then third one, dizono, in the name we can identify di, which means the pores which are present in the two layers in the middle position, that is the dizono. Then third one, that is panto, which are having so many so many pores are present in which are arranged in irregularly on the surface of the pollen grain. Now the third one I can explain, especially the pollen grain character. What is pollen grain character is nothing but the germpore, how they can show the character, which means which are having a round shape structure, either which are having a triangle shape structure, either which are having hexagonal structure, either colpate, either pore, both colpate and pore. This type of structures we can see in the character after the deposition. Now I can the characters, the first one, the pollen grains character, there is not at all character of pollen grain, that is C, that is stream C0. I cannot mention at the top position C0. And then second one C1, that is nothing but the pollen grain which are showing like a round like structure, this is called as leptome. Then third one, that is nothing but tricolpate. You know very well what is tricolpate, is nothing but which are showing three positions like triangle shaped structure, that is commonly called as tricolpate. Colpate, in the pollen grain, on the surface of the pollen grain we can observe colpate position. What is the colpate structure is nothing but we can observe like a spindle shaped structure on the surface of the pollen grain, that type of character that belongs to colpate position. Next one, the next one, that is this is the colpate porate. You can observe this is the colpate, we can observe as a spindle shaped structure right now. And now this is the common porate. Porate is nothing but commonly pore. pore which is present in the central side, this is the commonly porate. The next one, that is calpoporate. What is this is very important character for MSc. So many times they are asking in the MSc question. In the pollen grain, we can observe like carpet. Carpet is nothing but like spindle shaped. In the middle, spindle shape which are having like a round shaped structure. Round is nothing but that is porate, and spindle shaped is also present, that is commonly called as carpet. Both characters which present on the surface of the single pollen grain, that is commonly called as calpoporate. Now finally, that is poroporate. Poroporate is nothing but the which pores are present, the one pore which is surrounded by the another pore. This is the common poroporate. Why? According to advanced scientist, which is classified into characters, stream. Stream is nothing but there is no character. We can identify it as C0 and C1, that is nothing but lepto, and tricolpate, that is nothing but C3, and which are we can see the tricolpate on the surface of the pollen grain. Next, colpate structure, that is the common spindle shaped structure we can observe on the surface of the pollen grain. This is the C4. And C5, that is pororate. Porate is nothing but the round like either oval like structure which are present in the central of pollen grain, that is pororate. Calpoporate, calpo and pore. Calpo plus porate, which means which contain two characters like calpo and porate. Both characters will present this type of pollen grain, that is calpoporate. So very important. The next one, that is poroate. Porate is nothing but that is commonly we can observe porate, which means each pollen grain which are having up to 4 to 5 pores, each pore which is surrounded by the again one more layer. These are the common NPC system by the scientist of Hedman. That is NPC means N means number and position, and character. Now I can explain the one more important character. This type of NPC system of advanced scientists belongs to only dicotyledon plants, not for monocot. See, this is influenced to on the dicots, not particular monocots. Why? Because all the dicots plants will applicable for NPC, right? NPC again I can explain the brief outline about NPC. Why? Because NPC is a very important for theory. Even in some cases they given 4 marks question also. NPC system was first discovered by Edmund scientist. NPC means number and position and character. See, and in the NPC system we can use as stream suffix in the end, and n a stream, which means there is no number, there is not at all force are completely absent, that is a dream. And n0 and n1, that is monotream, which contain only single pore, and n2 which contain two pores to the outside and lower side, that is ditream. Tritream, which means which contain three pores are present on the surface of the pollen grain. Tetratream, which contain 4 pores are present, and pentatream, that is 5 pores are present. N6, which means hexa, that is 6 pores are present, more than 6, which is called as polytream. And finally, that is anemotream, which means there is no so many pores are present and which are arranged in irregular arrangement. Then position, that is if there is no total position, that is P0. And second one, then the position which is present to the lower side of the pole, that is called as catatream. And the anaketa means the pores which is present both side of the poles, upper and lower, that is called as anaketatrim. And anatrim, which is the pore which is located to the outside of the pole, that is commonly called as ana. And zono, which means which is located to the middle position, that is commonly called as zonotrim. Next, dizono. What is the meaning of the dizono? Which means the pores which is present in the middle, that is commonly called as dizonotreme. And pentatrim, more number of the pores will present, that is commonly called as pentatrim. Now I can explain the final character, that is nothing but if there is no character to the pollen grain, that is commonly called as trim, either C0. If round, which is in the central, that is leptotrim, and tricolpate. Tricolpate is nothing but which are showing as tricolpate condition, that is the tricolpate. And colpate, which means the spindle shaped structure we can see on the surface of the pollen grain under microscope. Poro, which means round like structure. See, spherical structure we can observe on the surface of the pollen grain. And calpoporate, which contain both characters, and calpo and porate, which means both characters will present. Next one, poroporate. These are the common NPC system. I can explain the MSc questions from the paleontology. What is paleontology? They're asking. What is the paleobotany? The study of fossilized pollen grains, which is called as paleobotany. And we can observe the paleobotany, either fossilized plants and fossilized pollen grains. We can observe a particular institute, that is Bzc Institute, which is located at Lucknow. This is also very important. And now the exam with made of which type of product they are asking in MSc, that is poropollenin. Then intine, it is common cell wall with made of cellulose and hemicellulose. And next one, anther, we can observe how many layers are present, that is endodermis. And second one, middle layer, and sorry, endothecium and middle layer and tapetum. We can observe three layers under microscope. And inner side, which are present the microspore mother cell. This microspore mother cell under participating microsporogenesis. What is microsporogenesis? Which means the microspore mother cell, which contain two set of chromosome, which is participating meiosis division, which producing four microspores. These are tetroids, which contain four cells. These are all applied cells. Why? Because which is participating only meiosis division. Now this is also very important. What is the function of the tapetum? They are asking what is the function of the tapetum. Is nothing but which are helping in the nutrition, either which are given food material to the developing pollen grains to the inner side of the anther. Next, what is dithecus and monothecus? At the time of the families, very important character and anther character. Either dithecus, which contain two rooms, and monothecus, which means which contain only one. The next one. What is ornamentals on the surface? We can observe different type of ornamental spines and so on. So next, what is the germopore, the function? Germopore, then we can identify on the name germopore, which means germopore, which are helping in the process of germination of the pollen tube at the time of fertilization process. Right now, the next one. What is the what is the condition of the male gamete, which are having only single set of chromosome, that is haploid in nature? Why? Because before fertilization process, both sex organs, like male sex organ of pollen grain, either a male gamete, either female sex organ, either female gametophyte, either egg cell is are having only a single set of the chromosome, haploid in nature. Why? Because before fertilization they are having only single setup. After that, after the fertilization, they can, the fusion of male and female gamete, the male gamete N and female gamete N, both are fused with, which is formed diploid cell, 2N cell, that is zygote. This zygote is directly converted into embryo. After the zygote is directly germinated to give a new plant body, that is diploid plant body, sporophytic plant body. So very important, sporophytic plant and gametophytic plant body. This is also very important. Next, I can explain. What is the function of the ornaments on the spines in the hibiscus? That is the protection. The spines which are helping the protection. Next, Who has proposed NPC system? That is common head domain scientist. What is NPC? They are asking NPC abbreviation. N means number and position and character. They are asking calpoporate. Calpoporate, which means which are having two characters on the surface of the pollen grain, like spindle shaped structure and the pore. Jono, they are asking Jono. Jono, which means that is also the pores, which is position of the pore which are present in the middle. Dijono, which means the pores which are arranged in the two rows in the middle position only. This is the NP system, even NP system and also MSc questions. Thank you students. Thank you one and all.